Assalamu alaikum my dear student, this is Miss Aisha Anwar with O-Level Biology Syllabus 5090. I would like to welcome you all in my academy, that is Biotic Academy. Here we are going to study and discuss about the O-Levels Biology Syllabus, the learning outcome of the syllabus. So today I will begin my class with this basic chapter of the syllabus, cells. Cell is the basic structure and function unit of all living organisms, and in this chapter we will be studying about the different organs of the cell, how they are performing their role in order to bring about the specific function, a specific job by the cell. So as you all are the biology students, so you are very much familiar with this concept of unicellular and multicellular. What are unicellular organisms? These are the organisms which are composed of single cell, and the multicellular organism are the organisms which are composed of more than two cells, 2, 3, 4 complex number of cells. As the number of cells could be increased to the giant organisms, the complex organism. But the question arises here, as we are saying that unicellular organisms are the organisms which are composed of single cell, like bacterial cell, which are very small and we cannot see it with the naked eye. So how we are able to understand the structure of cell, how we are able to understand about the internal organs present within the cell? So what are the gadgets which are required, what are the instruments which are required to study the internal structure of cell? For this basic understanding of the cell, scientists were capable enough to study the internal structure of cell with the help of this instrument, that is an optical instrument which is known as microscope. You all must have seen this diagram of microscope, and you must know about the function of this microscope. Okay, what is microscope and how it is working? Microscope is basically an optical instrument which is used to observe the organisms which are not visible with the naked eye. Means ke small organisms can be seen with the help of this microscope. So in order to observe this small organism or unicellular organism under the microscope, first of all we should know the operation of this instrument, how it is working, how it works in order to bring about a clear picture, a clear image of the unicellular organism. This instrument composed of different parts, so I would like to explain you the function of different parts of this instrument. We will begin from here. The eyepiece. Eyepiece is basically the place which is having a lens over here. It is having a lens. Another lens is placed over here, the objective lens. These two lenses together they bring about a magnified image of the small structures, or you can say it basically enlarges the apparent size of the organism. Apparent size of the organism means that it will magnify the image of the organism, not the organism itself. This is the body tube where the image form, and these are, you can see there are three objective lens over here, and by rotating this objective lens according to the need of our vision, we can adjust the image of the organism accordingly. It is having 5x, 4x, 100x, different value of the magnification lens are there present over there. Objective lens are present over there. Now this is the stage where we used to place the slide of the organism which we want to see under the microscope, and this is the condenser. This part is called condenser, which is used to adjust the light ray in order to see the clear image of the organism, clear image of the specimen, whatever we want to see under the microscope. Now here is the source of light. It could be a mirror or it could be a bulb, so it is depend upon the variety of the microscope which you are using. This is the base which helps to maintain the proper alignment of this optical instrument. Now these are the two adjustment knobs you can see over here: coarse adjustment and fine adjustment. Coarse adjustment and fine adjustment together helps to bring the fine image of the fine image of the specimen whatever you are observing under it. So these are the basic parts of this optical instrument. Now while discussing the working of this optical instrument, we use two terms, that is magnification and resolution. What is magnification? It is the process of enlarging the apparent size of physical size, not the physical size of something. As I told you that under the microscope we observe the enlarged apparent size of the organism, and that is basically we are using term magnification for that enlargement. And in the second heading, magnification of that or enlargement can be seen easily if it is having a proper resolution. What is resolution? Resolution is basically the term which is used to enabling the person to identify the minimum distance between the 2 points. Like for suppose I have put it 2 points over here. Now some of you may see that it is 2 separate points, and some of you may have this problem that it is a single point. Though the resolution of the eye will help you to see or understand the things which are very minute and they are not visible clearly, but under the microscope we can view these small structures, small details within the cells. So by having a proper resolution, it will be able to understand whatever the organelles or whatever the molecules present within the cells, how they are working, how they are moving from one place to another place within the cell. So these are the all things which are being cleared with the help of the proper resolution of the microscope. So it is basically the resolution which helps to give us a clear picture. Magnification and resolution are the two different things. Now in order to view the specimen under the microscope, we need to follow a step where we are going to follow proper procedure for making the specimen ready to be observed under the microscope. Here, according to your syllabus, you have to observe the onion epidermal skin. You need to have a slide, and then over the slide, first of all you need to peel off the skin, the thin skin of the onion, which is somewhat you must have seen, that's like very tissue paper like structure. Is basically it is like this, and if you observe it, it's like this, it will be appearing like this. This is basically the fine layer present between the scales of the onion, and these are basically the tissues of the onion. So according to your syllabus, you need to prepare a slide to observe this onion epidermal skin under the microscope. So first of all you will peel off that membrane layer, and then you will take a slide. You will put a dye over it, either methylene blue you can use. These are the basically dyes which are basically the staining method in order to make the slide, the colorless tissues, more visible, because you know that these are the colorless tissues, so they need a proper stain in order to get properly viewed under the microscope. So I'll put a drop of iodine or methylene blue, whatever dye we can use over here. I'll put a drop of dye over here, then I'll put the skin of the onion epidermal skin over here on the tissues, and then I will put a cover slip. It's a small square piece which I'm going to put over the slide, over that skin of onion epidermal skin, and then I will cover it, and then I will observe it under the microscope. Now when I will observe that slide under the microscope, it will appear like this. It's like plant cell will be appearing like this, and animals, if I am observing for suppose cheek cells, cheek tissues I have taken, and I have made the slide of that cheek tissues. So after the staining techniques, after the procedure, same procedure which I have told you previously, we are going to observe the animal cell. It would look will look like this, and the plant cell, the onion epidermal cell skin is going to look like this. You can see the rectangular cells which are present, and they are showing their nucleus. You can see the nucleus over here. So this is how you will be observing the plant cell, rectangular shape, and somewhat like this irregular shape of the animal cell you can observe under the microscope. So after observing these structures, if you want to go further in detail of these structures, you may use compound electron microscope. Electron microscope, what is electron microscope? Electron microscope is a microscope which is going to magnify your image, which is going to increase your resolution 1000 times more than this compound microscope. Its magnification power of compound microscope is 250,000 times x. Means it will show you the detailed structure within the cell, within the nucleus, the DNA and all other structures which you have studied in your syllabus. These are the all structures which are visible under the electron microscope. And while observing these structures, we used to take the photographs of them. If I'm observing a structure under the compound microscope, which is also a light microscope, we can say light microscope also. If I'm taking a photograph with the help of the light microscope, that will be called as photomicrograph. If a photograph which is taken with the help of the light microscope is called as photomicrograph. But if a picture which is of any specimen which I'm taking while observing it under the electron microscope, then it will be called as electron micrograph. Fine. So these are the terms you must have seen in your syllabus, that photomicrograph and electron micrograph, these are the differences between them. So now this is the actual picture of the electron micrograph and photomicrograph of this animal and plant cell. The pictures which are visible under the microscope, these are like this. And the picture which you have seen up till of different animal cell and plant cell, mostly they are the drawings, but this is actually it look like under the microscope, plant cell and animal cell. And you can see the organelles which are visible under it, under these cells, like chloroplast, cell wall, plasma membrane, Golgi apparatus, nucleus, mitochondria, vacuoles. And these are the all organelles and structures which are present within the plant cell. And in case of animal cell, you can see very clearly the mitochondria. Plasma membrane is the same as the cell membrane. Fine. Plasma membrane and cell membrane is the same thing. Centriole are present near the nucleus, which have their specific job for cell division. And Golgi apparatus are also there. Lysosomes are there, and nucleus is present. In animal cell, you can see it's there, the dark stain. So these are the actual pictures of these animal and plant cell under the microscope. Now we would discuss these different organelles, different structures of the plant and animal cells one by one. So first of all, you should know what are organelles. Organelles are basically the tiny organs which are present within the cell, and they each have their specific role within the cell. Within the cell, mitochondria, Golgi apparatus, endoplasmic reticulum, ribosome, these are the different organelles which are having their specific job, and the proper functioning of these organelles bring about a collective function of the cell. Altogether, the function performed by an individual cell is basically the outcome of the function performed by different organelles within the cell. So I hope this is clear to you. Now organelles, first of all, we'll begin with the ribosome. And ribosome is basically a very important structure within the cell. Its function is to perform the process of photosynthesis. Ribosomes are present in prokaryotes, eukaryotes, both the type of cells they are present, and their function is to perform the job of protein synthesis. And we will be discussing about the process of protein synthesis in detail in the later on in the next part of our syllabus. So the main function of ribosome is protein synthesis, which is a very important function because protein is much needed within the cell for different cell activity, like for making different enzymes and hormones and all cell membrane. These are the all different tasks which are being accomplished by this ribosome, and it performs this function of protein synthesis to be utilized in different function within the nucleus and outside the nucleus. In the cytoplasm protein is needed, and this ribosome basically helps to perform this job. Now the next come mitochondria. As we are in the beginning of the syllabus, so we will not go into much detail of their function, because later on in our syllabus we will be discussing them in more detail. Mitochondria is the nest organelle which is present within the cell, and that is the site of energy, and usually we say that it is the powerhouse of the cell. It releases energy in the form of ATP, which is basically the currency of the cell. Means cell use this ATP to perform different metabolic reaction. So mitochondria basically generate this energy, this ATP, by the breakdown of glucose molecule in the presence of oxygen. That is basically the process of cellular respiration. So mitochondria is also a very important organ within the cell, and it has to generate the energy for the cell metabolism, so that cell can conduct its performance, can conduct its metabolic reaction smoothly. Now after mitochondria we'll be discussing the next, that is the nucleus, which is basically the, we usually say that it is the brain of the cell. Nucleus is the brain of the cell. Why we are saying that it is the brain of the cell? Because it is controlling and regulating the activities performed within the cell. Means that there should be someone who is controlling whatever going on within the cytoplasm, within the nucleus. Even the transfer of material inside the cell and outside the cell, endocytosis and exocytosis, these are the different terms which we use for the exchange of material within the cell. So these are the all processes which are being controlled by the nucleus, and nucleus perform this job very well. For example, you can see that here I have mentioned that growth and metabolism, and it basically responsible to carry the hereditary material within it, the genetic material within it, which is controlling the physical and biological basic makeup of the living organism. Now the cytoplasm is basically the jelly like fluid which is present outside the cell. Means ke your cell has nucleus, and outside the nucleus there is a jelly like fluid which is holding all the organelles within it, and it provides a platform for the metabolic reaction to occur. So this jelly like fluid is called cytoplasm. That is a mixture of water, salts, protein, glucose. These are all the things which are present in this cytoplasm, and it not only provide the platform for the metabolic reactions to occur, but it also helps the cell to maintain its shape to a certain extent, and protect the cell from the outer environment, help to give it a proper, you can say, a better shape, because it is not having the animal cell. If I'm talking about animal cell, it is not having any cell wall which is going to maintain its shape. So the shape of the animal cell is basically based on this cytoplasm. After that we'll be discussing about the cell membrane. Cell membrane is a very thin layer outside the, you can say, it's the boundary of the animal cell, and it's the second layer in case of plant cell. So cell membrane is basically a gateway to keep a check on the exchange of material, which is are the material which are coming inside the cell, what are the material which are going outside the cell. So these are the things which are being controlled by the cell membrane. So you can see over here that it is the second layer over here, and the cell membrane is the first layer in case of animal cell, and its function is to keep a check on the exchange of material. It is the material which are going inside and outside of the cell, and also it helps the cell or protect the cell from the transport of the toxic material inside it. कोई toxic material cannot enter inside the cell, and it helps to remove out the toxic material out of the cell. Now the second one is here. You can see cell wall, which is not present in the animal cell but it is present in the plant cell, and it helps to give a very proper shape to the plant cell, which helps to give the strength to the plant cell, because you know that plant is not having any skeleton within it. So how the trees are standing in a erect position with the help of the roots and with the help of this cell wall along with the turgor pressure. These are the different factors which help the tree to stand in an erect position, and they maintain their shape and they maintain their position in the external environment, and that is very helpful for the plant. So here we were discussing about the structure of the plant and animal cell, and you can see that in case of animal cell, you can see nucleus is present in the center of the cell, and in case of plant cell, it is present on the side of the cell. Why it is present on this lateral side? Because of the presence of big vacuole within it, and this vacuole is responsible to store nutrients, minerals, water, and the content of this vacuole is called cell sap. That is very important with reference to the turgor pressure, and helps to maintain the shape of the plant body. And chloroplast, you can see in this plant cell there are many chloroplasts within it, and the function of this chloroplast is to trap sunlight for the process of photosynthesis. Basically this organelle contain a chemical that is chlorophyll, which is needed to trap the sunlight for the process of photosynthesis. Now after this we will be discussing about the vacuole and chloroplast within the plant cell, because these are the structures which are not present in animal cell. They are only specifically present in the plant cell. Chloroplast, as I've told you, that it's a site of photosynthesis. This is, you can see it's LS of the chloroplast, how it looks from inside. It is having a thylakoid layer structure, which are basically thylakoid layer are basically the site where the process of photosynthesis take place. And this chemical which is present within this chloroplast, the chlorophyll, this helps to convert the sunlight energy into the chemical energy for the process of photosynthesis. And one interesting fact is that this chloroplast contains its own DNA. It is basically a discovery that the scientists came to know, the biologists came to know, that these organelles are independent in a way, that chloroplast and mitochondria, these are the two organelles which are having this interesting discovery about these two organelles, that they contain their own DNA. Now here we can see vacuole, and that is basically, I have told you that it is called cell sap. The fluid present with this is vacuole, and this cell sap contains dissolved substances, sugar, mineral salts, and amino acid, which are needed by the plant cell during this processes of metabolism.